Dear students, welcome to the second lecture of Introduction to Microbiology. Let us have a small recap of what we have learned in our previous lecture. First thing, we have learned what is meant by microbiology. Microbiology is the study of microscopic organisms like virus, bacteria, etc. They may be unicellular or multicellular. Then we have learned about applied and industrial microbiology. Applied microbiology deals with enzymes related to unicellular and multicellular organisms and also prokaryotes and eukaryotes. Industrial microbiology deals with commercial applications of microbes in producing various products and also various applications of microbes itself in industries. Two main applications of industrial microbiology is first one is about fermentation, how to produce various products using fermentation, and the second is about controlling pollution and managing solid waste using microbes. Then we have seen various products produced with the help of microorganisms. The first one is milk and milk products. We know that milk tend to spoil faster, so milk has to be converted into various other forms like cheese, yogurt, kefir, etc. so that it can be preserved for a longer time. We know that all these products are produced in a large scale. Except for cheese, all the milk products uses the bacteria in the milk itself for fermentation, whereas in cheese, fungi is used. For all these products, milk is pasteurized and then fermented. Along with the products, many taste and flavor substances like diacetyl is also formed. The next product is yogurt, which is also a milk product. It is produced with the help of bacteria lactobacilli. To increase the protein content, condensed milk powder is added to the milk to be fermented. This is done for industrial production of yogurt. After boiling the milk and warmed to room temperature, bacterial strains like Streptococcus thermophilus and Lactobacillus delbrueckii are added in the proportion 1:1 for fermentation. Various fruit juices can be mixed with the yogurt to get different types of flavors. To increase the probiotic activity and shelf life of the yogurt, milk is pasteurized. The next product is butter. There are two types of butter, sweet cream butter and culture butter. Sweet cream butter was produced from the fresh cream of milk, whereas cultured butter can be got after fermentation of milk with microbes. Cheese is one of the main commercial products of milk. After performing some chemical and microbial test, some colors and microbes are added to impart sourness to the milk. After the milk getting fermented, the watery part is removed from the yogurt. For this, earlier an enzyme called rennet deriving from the alimentary canal of cattle were used, but now instead of rennet, the enzyme protease derived from fungi is used to make vegetarian cheese. After removing the watery part, cheese is processed, cut into pieces and left out for ripening. The period of ripening imparts different flavors and colors to cheese, and hence various types of cheese can be made. The freshly made cheese like paneer and mozzarella are always soft, and ripening will make them harder and increased in flavor. The last we have seen about probiotics, which are milk products which help in increasing the population of good bacteria, which helps in digestion and also decreases the harmful bacteria. Probiotics are now used to treat diseases like diarrhea and also in poultry. Now let us see our homework question. Your homework question was to learn about different types of cheese. I gave you a hint that there are 7 types of cheese. You can see the 7 types are given in the slide. The difference you see is that they differ in the fat content. We know that ripening of the cheese breaks down the lipid and protein content of the cheese. So depending on it, the fat content of the cheese also varies. The 7 times as mentioned in the slide, you can see fresh, aged fresh cheese, soft, semi-soft, hard, blue cheese and also flavor added cheese. Out of this, blue cheese is made by imparting the fungi penicillium, which gives a blue color spots in those types of cheese. For flavor added, many different types of spices and flavors are added to give a peculiar taste to those cheese. Let us continue with the products produced using microorganisms. Now we are going to see bread. We know that bread is made from flours of various cereals like wheat, ragi, etc. Nowadays we are getting multi-grain bread also. For making bread, first a dough is made with mixing flour, baker's yeast, salt, water and other necessary materials and left out so that the dough rises. What makes the dough rises is the fermentation of carbohydrates present in the cereal by yeast. This converts the sugar present in the carbohydrate into alcohol and carbon dioxide. The alcohol produced is especially ethanol. The yeast, baker's yeast, which is commonly called, is a fungi called Saccharomyces cerevisiae. Commercially available yeast are available in compressed dry form and also granular form. Along with the yeast, compounds like carbohydrates, proteins, vitamins, minerals are also added to enrich the nutrient content of the food we make using the yeast. Not only bread, many other products like vinegar, soya sauce, monosodium glutamate, which is commonly called as Ajinomoto, are all produced by microbial fermentation process. The sponginess we see in bread is due to the trapping of carbon dioxide in between the dough. The next product we are going to see is vinegar. Vinegar is used worldwide to impart sour taste to food and also for preservation of food items like pickle, ketchup, chutney, sauce, etc. Vinegar is nothing but 4% acetic acid. How vinegar is made? We know that during fermentation of carbon compounds like fruit juices, syrup, sugar molasses, starch, etc., we are getting ethanol. This fermentation is done by using the yeast Saccharomyces cerevisiae. After getting ethanol, microbial strains like Acetobacter and Glucanobacter are added which degrades this ethanol. Acetic acid and other similar products are obtained due to this degradation. The acetic acid produced is separated by a process called rarefaction. This rarefaction is nothing but reducing density of compound and extracting them accordingly. Then the acetic acid obtained is bleached with the help of a chemical called potassium ferrocyanide. It is then pasteurized. Then very small quantity of sulfur dioxide gas is mixed so that we can get vinegar. The product soya sauce is also produced by fermentation of mixture of flour of wheat or rice and soya bean with the help of fungus Aspergillus oryzae. This table gives you information of producing various types of beverages using microbes. The first one is Caffea arabica, which is nothing but our coffee. The microbe used is Lactobacillus brevis. Where it is used to separate the seed from the fruit. We are actually using the coffee seed. For separation of the seed, this microbe is used where we can get the beverage coffee. The next one is Theobroma cacao, which is nothing but cocoa. The microbe species used are Candida, Saccharomyces, Specia, etc. The process is same that of coffee where the seed is separated from the fruit. The next famous product is wine produced from grapes using the fungi Saccharomyces cerevisiae. We know that this is the yeast used for fermentation of the grape juice which gives wine. The last, using the fruit apple and yeast Saccharomyces cerevisiae, we can ferment it and we can get a product called apple cider. Cider is an alcoholic drink made by fermenting apple juice. So through this slide, we understood how beverages are made using microbes. We know that applied microbiology is the study of enzymes. Now let us see in brief where microbial enzymes are used. We know that in our digestive system there are various enzymes are secreted. Can you tell what are the functions performed by enzymes? These enzymes help in breaking down the complex food into simpler nutrients so that the nutrient can be absorbed effectively by the blood and the waste can be discarded. Now, can you name or give names of some such enzymes? There are many enzymes involved like protease, lipase, trypsin, tyalin, amylase, etc. Each enzyme is responsible for breaking down a particular compound in the food. For example, pepsin breaks down the protein, lipase breaks down the lipid part, that is fat in the food, etc. Let us see some other applications of microbial enzymes. We know that in a chemical industry, a catalyst is used. Catalyst is a compound that speed up a chemical process without actually getting involved in the reaction. If we are using chemical catalyst in chemical industry, there are lots of problems which gives unnecessary byproducts. The chemicals can corrode the wall of the container, etc. So nowadays, instead of using chemical catalyst, microbial enzymes are used for the same process. These enzymes are active at low temperature, pH and pressure due to which It saves lots of energy. It not only saves energy but also the corrosion of vessels is minimized or there is no corrosion at all, and as the enzymes are specific, no other unnecessary byproducts are produced. As there are no unnecessary byproducts, the process of purification of the product is highly minimized. These are the applications of microbial enzymes in chemical industry. In case of any enzymatic reaction, elimination and decomposition of waste is avoided and the enzyme can be used again and again. Hence, these enzymes are eco-friendly. Let us see some microbial enzymes. First one is oxidoreductase. These types of enzymes transfers electron from one molecule to the other. The next is transferase. The transferases transfer functional group from one molecule to the other. You will learn elaborately about what is a functional group, what are its properties, etc. in carbon and carbon compounds chapter. Next enzyme is a hydrolase. Hydrolase helps in breaking a long compound into smaller one. Also does the same job but uses a different method. The next isomerase enzyme. Before knowing the function of isomerase enzyme, you should know what is an isomer. An isomer is a compound, if for example you take two molecules which are made up of same atoms but the arrangement will be different. If the arrangement is different, they will have a different purpose. Those two molecules are called as isomers. This enzyme isomerase changes one molecule into the isomer of the other one. The enzyme ligase is used to join two molecules. As you have seen, these enzymes have a particular peculiar function so that only that particular function can be made using these enzymes, hence reducing unnecessary byproducts. The other uses of enzymes is like in detergent where the dirt or murk is nothing but a stain occurs at low temperature also due to the mixing of enzymes. This enzyme and detergent together removes this dirt. Glucose and fructose syrup, they can be obtained from corn flour by the action of enzymes from bacilli and streptomyces. Microbial enzymes, they are also used in various industries like cheese, we know that very well, plant extract, textile, leather, paper industry, etc. We understood that microbial enzymes have wide range of applications. We know that the food items like cold drinks, ice creams, cakes, juice are available in various colors and flavors. Do you think that these colors and flavors are obtained naturally from fruits? Find the answer. The ingredients printed in the bottle of cold drinks, juices and ice creams have a different meaning. You can see natural identical flavors and various different numbers and alphabets used. Find what they are for and what they denote and where they are actually derived from. To impart taste and flavor, many organic acids like glutamic acid, citric acid, which gives us our taste, are used. Microbes are used for production of these organic acids. This slide and the table gives you a detailed view of what are the items acids are produced using what kind of microbes and what is the source from where they are produced. The first is sugar and beet molasses ammonia salt using Brevibacterium or Corneobacterium. The amino acid glutamic acid is used, which is used in the production of Ajinomoto or monosodium glutamate. From sugar molasses and salt again using Aspergillus niger. Citric acid is used, which has wide range of application in drinks, the toffees, the candies that has a sour taste. The citric acid is the reason. Then glucose corn steep liquor, which has a high sweeter taste, is also produced with the help of this microbe Aspergillus niger, where gluconic acid is produced, which is used in the production of minerals used as a supplement for calcium and iron. From sugar molasses corn steep liquor using Lactobacillus delbrueckii, lactic acid can be produced. This lactic acid is source of nitrogen in production of vitamins. Then again from molasses and corn steep liquor using Aspergillus itaconus, itaconic acid is produced. This acid has industrial application like in paper industry, textile, plastic industry and also in gum production. These are other industrial application of enzymes. The next product we are going to see is this xanthan gum. Where is it derived from? This gum is obtained by fermentation of starch and molasses with the help of Xanthomonas species, hence the name xanthan gum. Now, where it is used? This gum imparts thickness to ice creams, puddings, milkshakes, instant soups, etc. It is variously useful due to properties like its solubility in both hot and cold water, its high density, etc. It is also used in the production of pigments, fertilizers. Pigments are colors, we know. Fertilizers, weed designs, textile colors, toothpaste, high quality paper, etc. Let us see some other substances obtained by microbial process and what are their uses. First comes citric, malic and lactic acids are used to impart acidity and sour taste. Acids, acid like glutamic acid, lysine, tryptophan are used in binding protein with other molecules. The products niacin and natamycin are microbial restrictors. That means they restrict the growth of microorganisms. Vitamins, vitamin C, B12, B2, they act as antioxidants. That is, they restrict the use of oxygen so that a food item can be preserved and also for production of vitamins. Products like beta-carotene, lycopenes, xanthines, luteins are useful in making edible colors. Polysaccharides and glycolipids are used as emulsifier. Emulsifier means if two liquids which are normally immiscible, that is they will not mix with each other easily like oil and water, using this emulsifier those two liquids can be mixed up. Then vanillin, ethyl butyrate which has a fruit flavor, peppermint flavor, essence of various fruits and flowers are used to impart that particular essence in food products. Products like xylitol and aspartame are used as low-calorie artificial sweetener in many food products. Let us summarize what we have seen in today's lecture. First, we have seen the production of bread using baker's yeast Saccharomyces cerevisiae. Then, how vinegar, soya sauce, etc. are produced. We have seen how microbes are helpful in producing various beverages like coffee, cocoa, wine, cider, etc. Other applications of enzymes produced through microbes in chemical industry, other industries, etc. We have also learned how commercial products are produced with the help of microbes like glutamic acid, citric acid, gluconic acid, lactic acid, itaconic acid, etc. We have also learned about how xanthan gum is produced and where it is useful. We have also learned how the substances obtained by microbial process are having various applications. Let us do an activity. Read the ingredients and their proportion printed on bottles of cold drinks and juices and wrappers of ice cream. Find out the natural and artificial ingredients. You do this on your own and see what you know and what are the ingredients you do not know. Homework for this chapter is: what are the ill effects of Ajinomoto? Find the answer. The rest of the chapter we will continue in the coming lectures. Thank you.